Hi friends, so now I am back with my next video, and in this video we are going to talk about transcription in eukaryotes. So in my previous video I have talked about the prokaryotic transcription, in which we have talked about different factors, the RNA polymerase, and how it works for the termination as well. And in case of eukaryotes, some of the things are same, but there are some things which are a bit different. So I will explain you in detail how they work in case of eukaryotes. Please make sure to like, share and subscribe my channel. So transcription in eukaryotes it is much more complicated as compared to prokaryotes, because in prokaryotes there is only one single RNA polymerase involved in the transcription, but in case of eukaryotes there are three different RNA polymerase which are required, and it requires many more transcription factor as compared to the prokaryotes, and it needs a promoter, regulatory sequence, upstream regulatory region as well. And there are enhancers, there are repressors which can also regulate the initiation of transcription by RNA polymerase. So I was telling you that there are different polymerases of eukaryotes. So there are basically three different polymerase 1, polymerase 2 and polymerase 3. And all these three polymerases, they are involved in different RNA transcription. So like polymerase 1, it is involved in the transcription of the ribosomal RNA, the one which is involved in the which is a constituent of ribosome. Polymerase 2 is involved in the mRNA transcription, or the which is the precursor form is called heterogeneous nuclear or small nuclear RNA. And then polymerase 3, it is involved in the 5s, like different subunit like 28s, 18s, 15s. So in the third it's 5s rRNA, and also for the transfer RNA which is involved in the transferring the the codons for the protein synthesis, right? They provide the amino acids for it. So there are different polymerase, and the most important is polymerase 2, which is majorly involved in the mRNA transcription. And today we will talk about polymerase 2. So there are different processes as also in eukaryotes. In prokaryotes there are initiation, elongation and termination. In eukaryotes also most of the steps are same: initiation, elongation and transcription. And I will talk about the differences in both of them, and if you want to know the basic process, please watch my previous video of transcription. So there are several initiation factors are involved for efficient and promoter specific initiation in eukaryotes, and they are called as general transcription factor. And apart from general transcription factor, there are also other factors are required to promote the significant expression. They are mediator complex, DNA regulatory protein and chromatin modifying enzyme. So we will talk about the initiation. The process is a bit similar. So if you remember, in the prokaryotes, it requires a data box which was present as -10, like from the start side. But in eukaryotes, this box is present at -30. But in this case, this box is not sufficient for the polymerase to bind. After recognizes this place which is called the promoter. The promoter is a site where the polymerase bind and activate the transcription. So this place is called the promoter, where the polymerase is going to bind. So this box is not enough for the initiation. You require other transcription factor, which are TFIID, which is our transcription factor second D. So after recognizes this place, it binds with the transcription binding protein to this place. And once this binds, it recruits other transcription factor A and B, which again bind to these factors. One binds the downstream and one binds to the upstream. And now after this binding, what binds is the RNA polymerase 2 along with E, F and H. So transcription factor second H, it's the helicase. So if you remember, the function of the helicase is to unwind the complex, because if you remember in the previous, in for prokaryotes, it is a closed complex, and now what we need to form is the open complex, so which is done by the transcription factor 2H, which is a helicase which unwinds both the strands of DNA. So this complex which is formed with so many transcription factor and polymerase is called the pre-initiation complex. So as I have already said, transcription factor second H, it is involved in separating the double stranded to provide the polymerase to single strand DNA template, and the transcription factor and the polymerase to bind in a particular order, right, as I have explained you the order, and this complex is called the pre-initiation complex. An activator repressor can bind and activate and modulate the transcription rate, how it is happening. So now, and activator, as you know, the it can increase the transcription rate, and repressor can decrease the transcription rate. So now we have talked about the initiation part. Let's go to the elongation part. So the elongation is exactly the similar for as we have seen for prokaryotes, because now what we have done is we have formed this bubble, the open complex, with the help of RNase H, and now it will start to recruit like different ribonucleotides based on the sequence, right? If you remember, the template strand is the one which is used for the transcription, because this strand is 3 to 5 and RNA is formed from 5 to 3. That's why we are using this strand, which is called the template strand, and the strand which is not used is called the coding strand on the sense time. So as I have already told you that polymerase contains so many subunits, helicase, single strand binding proteins, as well as other things to carry out other functions as well. So, and the phosphorylation leads to, like, there are different phosphorylation of the C terminal domain, which like leads to the removal of the initiation factor, but add the factors which are required for elongation and RNA processing. And then it moves along the template strand, and the ribonucleotides are added in the same fashion as in the prokaryotic transcription. So elongation is almost the same as in the prokaryotes, and they are added to the like as the RNA polymerase move, it adds the ribonucleotide. Here you can see instead of T we have U in the RNA strand, and then forms the mRNA transcript. So as we have talked in the previous video, that in prokaryotes the termination is by the row dependent and row independent signals. In the eukaryotes it's a bit different. It proceeds in the polyadenylation and then termination. So once you reach at a point that there is a need to stop the transcription, there are different adenyl group adenine which are added to the 3 end of the mRNA, which is linked with the termination. So the polymerase C terminal domain tail is involved in the recruiting the enzymes necessary for the termination, and they are mainly carried out by two protein, which is called the CPSF or CSTF, cleavage stimulating factor. These are the factor which binds and initiate the removal of the strand from the double stranded DNA. So here you can see we have the RNA polymerase, and then the RNA which is coming from this RNA polymerase process, like the transcription. And here you have the different signals, right? The 5 prime is phosphorylated, the cap, and once it's happening, there is this poly A signals in DNA. This poly A signals is a signal for the termination. So when it is transcribed, you will see that there are multiple AAA coming, and this poly A signal is recognized by two factors, CSTF and CPCF. As you know, in the previous one we have talked about cleavage and polyadenylation specificity factor and cleavage stimulating factor. So they recognize that the multiple A signal in the RNA, and then what it causes is the RNA cleavage from the polymerase complex. So now CSTF is released and what we get is CPCF, and then bind another protein which are poly A polymerase and the poly A binding protein which binds to the different A signals like the A tail, and then it signals the different poly A binding proteins are added, and then this is the termination, because basically the poly A signals is a signal like in case of prokaryotes also, which is a row independent termination, right? The signals to cleave the RNA from the RNA polymerase and add different poly A binding proteins which binds to these signals. And that's it. And if you have any doubts, please let me know in the comment section. I hope you like this video, and please make sure to like, share and subscribe my channel. Thank you.